Uh, so I'm Brian Herm. I'm part of the NeMO team, and I'm hopefully going to give you some practical walkthrough of the NeMO analytics, building on what Mike and Owen have discussed about the ecosystem, being able to use the resources of the BICCN in a very practical way. And so if everyone can see my screen, hopefully, we're going to first talk a quick word about just the nitty-gritty about just the presentation itself. So we want to make sure that this is, in the age of Zoom, it's kind of a tricky thing to do a workshop like this. So the NeMO team will be monitoring the chats for questions. So that's one aspect of it. And the other thing you can do, if you, within Zoom itself, if you go down to the participants section, there's various icons that you can use for feedback. So if there is a portion, how I'm going to build this portion of the workshop, I'm going to go through a presentation first, and then we're going to go into the NeMO Analytics website and go step by step through some of the tools. So if at any point you may go faster, slower, if you have a question, either place it in the chats or use these icons. A few of the other team members will help me monitor that chat. And let's see here. Sorry, I've just completely lost my Zoom window. So this is a wonderful new age that we live in. Okay. All right, so as we just went over, it is staggering the amount of data that the BICCN has created over the past few years. And as we see, a lot of these are coming into publications at this time. We mentioned the archive, so there's a very general breakdown of data into genomic data that's being held by the NeMO archive. That's a lot of the data sets at the top, either epigenetic or transcriptomic data. You have imaging data being held and also electrophysiology data. And we're trying to build this ecosystem where users can draw upon all of this data and be able to really take advantage of the fact that we're trying to dissect the brain in many, many different ways at a very cellular level, understanding the connections between neurons, and building upon the wealth of information that we know about the brain already, its functions, the regionality of it, but now we can get down to the cellular level and really understand how neurons connect and how they act in specific regions of the brain. So again, you'll be seeing either they're published or they're just about to come out, a number of publications that are looking at, as a pilot, specific regions of the brain. So already we have a wonderful paper for cross-species looking at human, marmoset, and mouse. And again, that's looking at the motor cortex region of the brain. We also have one that takes a deep dive into the different modalities, the epigenetic and transcriptomic aspects of it. And if you're not familiar, since I'm going to be talking a lot in this workshop about single cell data, again, one of the major advances in the last few years is being able to get the exact gene expression profile of every single cell within, say, like a dissection of a slice of brain. And you can take that single cell data and go through and identify each cell type within that slice of the brain. And so you'll be seeing a lot of plots similar to this. This is either a t-SNE or UMAP, depending on the publication. And the various colors, this is a collection of each one of those dots is going to represent a single cell. And they're grouped together based on their gene expression profile. So as you work for that data set, you'll essentially cluster those cells together, identify the cell types. And from there, you can really understand what are the various genes that drive the phenotype of that cell, how similar are different cell types. The cross-species paper did a wonderful job of trying to match up, do we see the same cell types across species? Was there ones that are more species specific? And you'll also, throughout the next year, see a number of developmental data sets as well. How do neuron progenitors eventually develop into mature neurons? How does that process work out in various species and various brain regions? So there's an incredible wealth of data. And one thing that we're trying to add to this with NeMO Analytics is be able to go from the conclusions that are made in the paper and then allow the users to then take the next step. So take that same data, reanalyze it, and maybe compare it to other data sets, historical data, or even data that people have just generated. So that's kind of the overview. So all of those aspects, all those data types are within this ecosystem. Hopefully, you can bring it to the NeMO Analytics website and be able to essentially reanalyze it in a new way. All right, so again, how this is tied together, I mentioned before that we have the NeMO archive. We won't be getting into that too much today, but the idea is that all the genomics data is held there. You can search for it on various facets. So you can search by brain region, by lab that's generated the data, file type. There's both raw data as in FASTQ, all the way up through process data. And no, Owen was mentioning about collections. So for the manuscripts that are coming out, we're also trying to organize those files that are associated with a particular manuscript in these very dedicated landing pages. So for example, with the mini atlas data, essentially all that mouse data from the motor cortex, we have the different raw files organized by data type. So we have the different single cell data sets, be it 10x, the SmartSeq, and we also have the epigenetic DNA methylation and the ATAC data. And for each one of those data sets, we have the corresponding analysis files as well. So again, that's already a huge benefit in terms of if people really want to understand how the data was analyzed within a paper, you can go there, and that can be linked directly to a paper. The kind of, obviously the analysis side of it, you can have a link to that data within your paper and go right to that profile in NeMO Analytics. So I'm going to show you a couple of examples how we're supporting some of the BICCN papers that are coming out. This is just a quick example of taking that exact same t-SNE coordinates that are presented in the paper, and in NeMO Analytics, people can see, oh, they have a gene of interest. They can see where's that gene expressed in the various cell types. And this is the major part of my portion today. I'll walk you through how to look at these profiles and build these profiles. So again, these come hand in hand. So again, there's a wealth of raw data on NeMO archive. Currently, we're working, you'll on day 2, on the 27th, you'll be walking through the amazing power of Terra. So one wonderful aspect of NeMO archive, all of those files are also on a Google Cloud. So those files are instantly portable over to the Terra processing platform, which also uses Google servers for doing all its computation. So when you go to the archive, you don't have to download any of those files. All that storage is handled for you. You can move it directly into Terra, process it there if you wish, and they already have hundreds of really well curated pipelines for doing analysis. So already we're trying to make all that data very easily available to folks to analyze. There's also an ongoing effort to reprocess in a uniform manner all the data that's coming from the BICCN as well. So NeMO will be also holding a good deal of the process data as well. So that could be instantly moved over to NeMO Analytics. So anything that's at a counts level file, so again, for single cell data, that means you have a matrix of genes by cells, and we're going to go through a quick example how to do that later on. But those process data on NeMO archive, with all that rich metadata, can be ported over into NeMO Analytics and analyzed on its own, or it can also be compared to existing data sets. And again, we're going to go step by step for this. Again, a lot of the papers are on there. In the second portion of this talk, Jairam is going to go through how we use EpiViz for displaying epigenetic data in a genome browser view. And I'll also be walking you through some tools. So the idea is that you have the data presented in the paper. There's other ways to analyze that data, be it comparisons. There's many plots that you can create, and also gene expression differences that they can tackle. So a couple of examples, and this is also kind of a shameless plug. If you're publishing a paper and you want to host your data on NeMO Analytics, please reach out to us. So this is especially well suited for single cell data. So for example, from the MOP mini atlas data, we have the gene expression data, as I showed you before. We have the exact cell types that were derived from the paper. When you get to NeMO Analytics website, you type in a gene, you're shown the gene expression. And along with it, you can also display any other data modality. So here we have the epigenetic data presented in EpiViz. You have a very much familiar genome browser view. So we've typed in GAD2, here's GAD2 in the browser. You have the ATAC data from a couple of different inhibitory excitatory neurons, and you have the DNA methylation data as well. So that will track essentially now on a gene basis. In a similar way, in the cross-species paper, this is just an example of extending the data. So they did a great job of comparing each individual species. And this is just an example of, again, for a single gene, what is that gene expression in the different species across the different cell types? You can very quickly see, there's a difference. This mouse GAD2 in the L6 cells in mouse is a little bit higher than it is in human and marmoset. And you can dive then into the ATAC data, the DNA methylation data, and see, oh, okay, there's a difference in the open chromatin regions in this part that may explain the species difference there. So many different ways you can display the data. And soon, the flagship, let's see, just got the working through the reviews. We're building profiles for each one of those figures, and so there's a lot of flexibility in NeMO Analytics. This is actually taking advantage of using a scatter plot. And the MERFISH data is a spatial data where you can actually have, they interrogated about 250 genes per cell across a slice of the motor cortex. And they have the positional information of every single cell. And so what I'm doing here is just simply laying out a scatter plot of those cell positions, and I'm overlaying the cell type information. So you see the beautiful layering across the cortex. You can also display the gene expression. And so, for example, this gene, TEDM3, is specific to just about the layer 2 region. And we see it, again, displaying it in a different way. We have the violin plots summarizing that data per cell type. So the idea, again, taking a figure directly from a paper, displaying it in a new way, and allowing the user, so they read the static page, they go to NeMO Analytics, and they can explore that data set further. And another thing that's very specialized is being able to create SVGs. So if you want to augment a image that's within a paper, in this case now we're looking at patch-seq data, which both looks at the morphology of the neurons, the electrophysiology data, and also they've extracted the RNA from individual cells, and you get that gene expression profile. I've added onto, these are actually images from the companion papers, summarizing the gene expression per cell type. So essentially lifting that image from the figure and then adding onto it these little bars right here that act as a heat map, summarizing the gene expression per cell type. So, a lot of flexibility in building the profiles. All right, so again, if you're interested in supporting your paper on NeMO Analytics, there's only a few things that we need. It's just simply the counts files. If you have cell type metadata, this is something that's obviously very useful for NeMO Analytics. Going back to Owen's point about building this ecosystem, this is also something that we're trying to integrate into the archive as well, making that metadata available to everyone. But any and all cell metadata that can be made available into the plots, either TC or UMAP coordinates if you want to have that displayed right away, a faithful recapitulation of what's in the paper. And then if you wanted to match the colors that's in the paper, then that could be added in as well. And then what the user can do is we can actually have a link in the paper going directly to this, and then they can explore your data. So this is an example of how that link might work. You can actually have a very short URL added to the bottom of the figure. They can click on that and go right to the profile. They'll be presented with the front page. They type in a gene, and then they can explore that right away. So if they're reading the paper and maybe they're super focused on a single gene in a disease context, they can go right to it and see, oh, okay, I actually want to further explore this cell type because that's where it's expressed. All right. So for the remainder of the talk, I'm going to go through a couple of tools. Again, NeMO Analytics can tackle basic gene expression differences. So this is just an idea of the compare tool. If you have a data set and you have cell types already designated, you can compare two different cell types and get out of that list of top differences. So that's one tool that I'm not going to explore that too much, but this is the one we'll really get into, is the single cell workbench, which uses ScanPy program as a means of analyzing the data. And the big hope here is that we hope that this could be a good gateway for biologists who don't necessarily have the bioinformatics background, that don't want to get into the programming, but want to be able to reanalyze single cell data. We've taken every single step of that processing and presented it as a GUI. So they can go through and they can make all the proper filtering of that data that you would if you were going through the programming. So hopefully that opens up this data set to a wider audience of scientists. The other flip side of it is, even if you're a programmer and you would normally do this in your own coding, if there is some quick way of getting to the data set right away, this can also give you an avenue. If you didn't want to go and download the data to your own server and go through that rigmarole, you can access the data sets that are on BICCN, go through the analysis, and get a result much like you would if you were programming. And we're going to walk through this step by step throughout the workshop. And then this is a further extension. There's other ways of also identifying gene differences, which we'll explore. Okay, and just a quick thank you to the team. So before we dive into things, any general questions here? Okay. All right, if you would please navigate over to the NeMO Analytics website. And so that's just nemoanalytics.org. Make sure it's a HTTPS. And that's something where I've sent out the link in previous emails. If you have not created a login, if you come to this website and you see in the upper right-hand side, create account, it's pretty simple. Clicking on that, there's just a few fields to add in, and you're off and running. All right, so unfortunately, I cannot see the participant listing, I don't believe. So if that's, there we go, perfect. All right, so I'll try and watch out for if people are raising hands in the participant list or need help. Okay, so at this point, you should have a login for the website itself. I'll also be following along a couple of things. So this is where it gets a little bit tricky, flipping back and forth between the Zoom presentation and Windows, I understand. To add on one more confusion, the link I've put in the chat, this is the outline which we'll be going over today. So if you need help, there's a few links to where you might need to be in the website. There's also links to the manuals that also kind of walks you through step by step. So if you ever get lost, ask a question, refer back to this outline. There's many ways for us to help you. Okay, I'm assuming that everyone has a login and we're all set to go. Okay. So to give you an overview of the front page, if you ever need to get back to the front page, you can always click on the upper left-hand corner NeMO icon. So that'll always bring you back to this front page itself. In the upper right, you actually have a drop-down menu underneath your name. There's a few things we'll explore here. So you can manage data sets, you can upload data, which we'll practice as well. So that's a few items will always be up in that upper right-hand corner. I believe the links to the manual are here, and then you can always email us with questions. So as the website stands right now, you get to various data sets through putting in a gene symbol. So in this upper bar, there's a number of data sets that we've already curated. A lot of these things are reflected in the publications, like I mentioned as we are going through the presentation. We have a few mini atlas ones, both. They did an integrated analysis where they merged all the different data modalities, and then there's just simply the gene expression data. There's a few epigenetic data, so on and so forth. So as we build more and more profiles, either supporting publications or as we see that there's natural collections of data across BICCN, those will be added to these profiles. We also have a couple of extensions on NeMO analytics. So NeMO AD will be a new Alzheimer's link to this. So if you're also interested, I've mentioned that you can support your own publications here, but if you wanted to also reach out to us and clone this entire site for like a very specific purpose and other things, this was actually born out of the hearing field. Ronan Hertzano did a wonderful job pioneering the site for the purpose of exploring hearing data sets, but this has been adapted for the neuroscience as it stands right now. Okay, and then these are just a couple of tools that we'll get into a little bit later, but right now we're going to focus on diving into a profile. All right, so the first one we're going to practice with is the motor cortex. So the fourth one down, I believe, for hopefully everyone, motor cortex cross species. So we'll select that. And then you can just type in a gene. I'm going to type in GAD2, as it seems to be my favorite for these guys. Now, before you search, if you wanted to know you spell that gene right, you can click on exact match, and that'll get you to just that gene. Otherwise, it'll search for any gene that has that name somewhere within the gene name. All right, so you're now into a profile view. Again, the top is going to remain the same, so you can always get back to the home page. On the left-hand side, you have those genes. We have exact match turned on, so you only have one gene choice. If that was turned off, then you will see a list of genes that may have GAD2 in its name. At the top of the page, you actually have gene information up here. So we have annotation by organism. Right now we have mouse selected, and if you clicked on this drop-down right here, this gives you a little bit of information on the gene itself. So a bit of what Go categories it's associated with, and maybe a little bit of description in some other guys. And if you wanted to get, for example, human-specific information, you would click on the different icons, and then the drop-down menu gives you a little bit different information based on what is known in human or the other species. We also have a series of link outs. So again, these are going to be based on the gene itself. So if I clicked on any of these guys, it would give me more information on that gene itself. So if, for example, I clicked on UCSC Cell Browser, this actually pulls out the Kriegstein early developmental samples, and it shows me where GAD2 is expressed in that data set. So this just gives you an example of another way to explore other data sets based on that gene. So if that was your focus, you can really get a wealth of information. Okay. For the individual panels themselves, so each panel will be its own data set. And they're going to display a certain plot. We'll practice, there's many different types of plots you can create here. In this case, we're just recapitulating that reduced dimension space that was presented in the paper. Again, we have where the gene of interest is expressed. If you look at the top right of each small window, you have this drop-down menu. And there's a number of things that you can do. So first off, the top one displays. This is all the other plots that you may have made. I'll just click on this for myself just to show you. So this particular data set, I think there was also a bar plot that was created. So these were just different plots that were made. Right now, this middle one is selected as the default. That's what's going to show up in the profile, but you can switch back and forth between which one is the default here. View just simply zooms in on that image. Information, this is where we're going to have a little blurb on that data set. So hopefully a little bit about the experiment, how the samples were collected. In this case, it goes into detail about the various cell types. So that gives you a nice background of what that legend means. When you upload a data set, hopefully you'll present as rich a metadata information. Publication, if there's already a publication established, you can put that information into the metadata, and when you click on that, it'll go right to PubMed and right to that paper. Similar idea for the geo. So if the data is deposited in geo, it would take you right to there. We're going to add on in the future all the other archives within the BICCN so that you can link, if the data is in NeMO, you can go right there. The compare tool, I showed you a little bit about that in the presentation. So you can take a very superficial gene expression difference between any two of your cell types. So that's a dedicated tool for just finding broad gene expression differences between established classes within your data. But what we're really going to focus on for the rest of the time are two things. One is the cure rate, and that's where you can go in and change the plot type. So you can create scatter plots, bar plots, violin plots, and we're going to explore that tool a little bit later. And then analyze is that single cell workbench. So if I wanted to take this data set and reanalyze it, I can walk through all those steps, which we're going to try out. Okay, and the last thing, this pen tool right here, if you wanted to take a note on the data set, so say for example, you're exploring this and you wanted to jot down a note that, oh, okay, GAD2 is expressed in L6, you can write down that note, it'll save it, tag it onto that data set, and you can read it back to yourself later. All right, pause for a moment. Any questions on just general profile and how that works? Okay. If you would please, let's return to the front page. And we're back here. And now we're going to get into the super tricky uploading data. So if you haven't seen it yet, the outline that I put in the chat, we're right here at the bottom of the first page. And if you would please click on the example metadata file and the Excel formatted data set. These two little files are the ones that we're going to use to practice uploading data onto the website. So just follow those links. You can download those. Those are just on Google. As you're downloading that, I'm going to walk you through the files that are needed. So first off, this is the metadata file. You can certainly borrow this, put in your own values. The only thing we really need are the values in beige here. So a title, again, this is going to be a dummy data set. I've pared severely down a single cell data set from human development, and it's just a slice of the cortex. And so I'm just titling it a cortex example for workshop, very original. The summary is where you can put in that nice paragraph describing the experiment, describing the samples. And one other thing, there's a drop-down menu for single cell, for the data type. So in this case, it's a single cell RNA-seq data set. One very important thing for the function of NeMO Analytics is that we link all genes back to the ensemble ID so that it's unique across the data set. And so I've mapped all the genes for this example back to ensemble build 96. So that's probably one of the most important things for allowing your data set to function properly within NeMO Analytics. You can add in a little bit of contact information. And then the last important thing is looking up the tax ID for your particular species. This is human, so that's 9606. I believe the other common one, mouse, I think is 120, I believe. But the nice thing is, if you were confused, you can actually look up data sets within NeMO Analytics and look for examples. So that's the metadata page. The data itself, one format that you can give it is an Excel spreadsheet, but the principles apply to all methods of getting data up there. The most important part is an expression matrix. It's going to be genes in the rows and cells in the columns. So one thing that's always striking to people new to single cell data is how many zeros there are. Essentially what each column is representing is it's representing a cell, and it's the number of transcripts detected per gene. So we see, for instance, there's a ton of zeros for this first cell, and that's normal. So the nature of single cell data, especially the 10x data, you're only really capturing a sampling of the transcripts within a cell. So that's going to be the expression tab. The genes, that's my lookup between ensemble ID and gene symbol. And then lastly, the observations. This is if you have any cell level metadata. So we've kind of flipped the table a little bit. Now cells are the individual rows. These cell names must match up with what was seen in the expression tab. And since I've already played with this data set a little bit, I've analyzed it before outside of NeMO Analytics. I know what cell types are. And I also have got it to the point where I've analyzed the t-SNE, so I can put those coordinates right there in that cell metadata. All right, if there are no questions about the format of data, we're going to go, and hopefully you've downloaded those files by now. If you go back to the front page, we're going to click underneath where your name is in the upper right, and we're going to upload a data set. So notice that there's a difference between gene expression and epigenetic. Jairam in the afternoon is going to walk you through the epigenetic upload. I'm going to go through the gene expression right now. So just click on that. And hopefully you should get to a page that looks like so. And the first thing you're going to do is upload a metadata file. Again, if you're new to this and you didn't have that example I just gave you, this is an example of those metadata sheets. You can just fill in your own fields. So I'm going to click on Upload Metadata File. And I'm going to find on my computer where that is. So in this case, example Cortex metadata. Open that up. And this will quickly show you exactly what you had in that spreadsheet. And you just check over to make sure that all that's correct. Scroll down to the bottom and you click continue. So metadata is loaded up. Again, I was mentioning there's a few different ways you can package your data to upload the three important pieces: that expression data, gene information, cell information. That's you'll see examples of each of those types throughout here. So there's looks like three different ways we can put Excel data, MEX files, which come right from Cell Ranger, or you can also do a 3-tab tarball. So tab delimited files for each of those. And just a quick reminder, NeMO Analytics, it's not just single cell data. So you can upload any type of data you want at this point. You can put in, you can even do a microarray, like if you had genes by brain region or like however that experiment was set up. Anything that's genes by something can be uploaded onto NeMO Analytics. So you can still take that wealth of historical data and use it to compare to new single cell data, which a lot of times is very helpful for looking at these major differences, or maybe there's been a very careful developmental experiment that's been done. Comparing that to new single cell data is actually very, very helpful. So we're going to select that expression file that you downloaded before, example Cortex data, open that up. And again, this is a small file, so hopefully it shouldn't take too long. You can double check to make sure that it's the same dimensions that you know. So I just made up, again, a very simple example, 94 genes by 160 observations. In this case, that's number of cells. I'm going to push submit. And what this leads you to is actually that curator. So we're going to get into this in a moment, but this is where you could plot the data in many different ways. So this is just showing you a couple of examples. So we're actually going to skip this for right now. I want to get right to the single cell workbench. All right, so how are we doing at this point? Everyone's got their data uploaded? I'm seeing, I think we're good. Okay, again, just throw a question in the chat if you need help. So we're going to navigate back to the front page. So clicking on that NeMO, and we're going to get into the workbench right smack dab right there in the middle of the front page. And again, you can use the workbench with any new data set that you've added or any data set that's on NeMO Analytics already. So choosing your data set, hopefully it should be the first one that pops up. You're looking for a Cortex example for workshop. And again, that's why it's important to keep track of that title. And so hopefully this can also be a tutorial into what are the decision-making processes behind analyzing single-cell data. As a general overview, everything in the left-hand side is the steps that we're going to take as we analyze this data. And each step is released by a different toggle switch right here. So everything that you're working on is presented as a box right here in the middle. The first step is filtering either the cells or the genes. So some things that could happen in a single cell experiment, there's the phenomenon where there's doublets. So as you're processing your cells, sometimes two cells merge together and you see way more transcripts in that cell than you would expect. On the flip side of that, perhaps there's a cell that just did not get many genes at all detected in it. So that could be two filters. So you can say, okay, exclude cells that have less than 5 genes in them, and that's just way below, or cells that have way too many genes than I would expect.
Hey, Brian.
Yes.
So I just had a question sent to me, which was, is there a preferred browser that you're aware of?
Chrome is the preferred browser, yes.
Okay.
Yeah, thank you. Get that over here. Okay. Yeah, unfortunately, folks, we do all of our development on Chrome. I know that people have been having problem with Internet Explorer, so. Okay. And then in a similar way, genes, sometimes you may want to exclude genes that are only in a handful of cells, or genes that are in a preponderance of cells. So they may influence the analysis one way or the other. So this is just a way of filtering those. If you do want to apply the filter, you would click the box. You can type in the number, and then you can make that decision. If your data set's sitting there for a few days, there will be preliminary plots that are generated that kind of give you an idea of this distribution of genes and cells within your data set. All right, so I'm going to click on, I'm actually not going to do any filtering only because this data set's so small. It's just a test data set. So clicking on apply filters just to keep us moving here. And what this returns is just a nice display showing you whether or not any genes dominate your data set. If I wanted to try and get rid of this one, maybe I can filter out genes that are in more than 160 or 150 of my cells. So that kind of gives you a good sniff test initially to make sure that you have a pretty good distribution of genes. All right, to get to a next step, you just simply go through, starting from the top down. I'm going to click on that toggle switch, which makes the next box into view. Another way that people quality filter single cell data is by mitochondrial content. Sometimes if you have greater than, the magic number seems to be about 5% transcripts coming from mitochondrial is kind of the break point. If it's higher than that, sometimes the interpretation is that the cells have experienced damage as it went through the process of dissection through processing. So sometimes people filter based on high mitochondrial content. The kind of caveat with that is a lot of times with developmental data sets, you want to be careful that rapidly dividing progenitor cells are also not filtered out because they may seem to have higher mitochondrial content. So you could filter. My dummy data set actually does not have any mitochondrial genes, so we're actually going to skip this step, but you can filter based on mitochondrial genes in this step right here. All right, marching forward, the next thing we're going to do is identify highly variable genes. So oftentimes single cell data sets are thousands or tens of thousands of genes by tens of thousands of cells. And to analyze that massive matrix is very computationally intensive, and also you can usually get to the answer in a much reduced space. So there's two things we do. One is we reduce the number of genes by identifying those that are most variable. And this is this first step. So usually in a data set that has, say, 30,000 genes sampled, we may reduce that down to about 2,000. And there's a couple ways we can do it. We can use the same algorithm that's used in SROT or Cell Ranger. You can adjust those parameters here, or you can simply just say, give me back the top X number of genes. And again, you can say like 2,000, or because we're using this small data set, I'm just going to select all the genes within this data set. So I'm typing in 94, which is my whole data set for this one. Again, this is a little bit of an abstract thing, but we're just practicing here. And so what it'll show you is it's a nice scatter plot of the dispersion of the genes versus the mean expression of the gene. And usually if you're just taking the top variable genes, it'll be at the top points up here. Again, we're just, for the purpose of this practice, we're grabbing all the genes. Now, the important thing before you move on to the next step here, we have to click on Save the Genes. Okay, so now that's reduced our data set down from the full set of genes down to the most variable genes. So that's filter number one. The next step, principal component analysis. We click on that, and please stop me if you guys are, seems to be doing pretty well. The next thing that we want to try and do is reduce down essentially the cell information now. So we may have a data set that has 10,000 cells within it or 1 million cells within it. We can now take that information and reduce it down to principal components. So if we just click compute and plot for this one, we now go from a data set that's variable genes by cells to variable genes by PCs. And we already see that each dot rate in here is representing a cell. And even on the PC level, we're starting to see some structure within this. These cells are very similar to each other. These guys are kind of similar, but there's a little bit more variation within them. And we also see how much variance each PC is explaining over here. And so if we wanted to, at this level, let's see here, toss in a couple of genes that we may, are interested in the data set. These are just a few that I knew from before, marked various populations. We can see in this tiny little view right here that these genes are already delineating major cell types, astrocytes, oligodendrocytes, et cetera, et cetera. So this kind of helps you as we're marching along, kind of get a sense of, there's something about the data set, you're expecting certain cell types. You can check that along the way as you're making progress. This second one will show you what are those genes that are driving the top PCs. Okay, so if we calculate our PC, our data frame is now variable genes by PCs. We can now further cluster these guys. So there's very common use of t-SNEs or UMAPs. The idea behind this is that you're going to be essentially clustering genes by gene expression similarity. And here we want to type in the number of neighbors. Essentially, it's asking how big of groups of cells am I considering at one time. So for this small data set, I'm going to suggest just you doing 5 neighbors. If you have a larger data set, you can certainly expand that a little bit. And number of principal components that we're considering, let's just take the top 10. And as a very high level difference between t-SNE and UMAP, t-SNE really focuses on what's a similar grouping and place them together. UMAP takes that a little bit step farther and tries to keep a relationship between those clusters, as in what the similarity is between them. People are now favoring UMAP. So I'm going to just plug that guy in. So this is a classic view that you see all the time in papers now, how single cell data is represented in either this t-SNE or UMAP. And again, this starts to describe how similar the cells are together. And we see that right there. And we see that it's also concentrating all those cells that were more scattered out in my radioglial populations, and now they're a little bit more uniform. And that neuron cluster is still together. Although now we're starting to see a little bit of structure within it. And just for kicks, let me just show you what those genes look like again. This is, again, our neuron cluster down here. This is a little grouping of inhibitory neurons. And so, as you're working through this, use your biology mind of starting to tease out what these cell types are. Okay. We're good. How many cells can you tool work with? It certainly handles tens of thousands of cells pretty well. If you have a specialized use case, we can always reach out. It's sometimes with the computational limits, all we have to do is kind of ramp up how much resources we're asking from Google itself. So if there's a special data set that we want to process, we can certainly work with you on that. But generally right now, the data sets in the tens of thousands work very well with this tool. Tens of thousands of cells. All right, clustering. So this is where we look into the data set and see what are the natural clusters of cells. And this resolution number, starting with 1.3 is kind of arbitrary. This is, again, as you're looking at this as a biologist, what kind of makes sense in terms of clustering is starting to reflect the cell types that I'm expecting out of the sample. For my purposes here, just because it's a smaller data set, I'm going to bring this down to 1.5. And when you lower the resolution, you're going to get fewer groups, fewer clusters. All right. All right, so there was a question about the PCA, if there's an error. Oh, for this, I think there might be a thing about plotting the top genes. There might be an issue with, sorry for, I don't know if I should be responding individually to the question. This image right here, there's somebody actively working on this display. There's something that popped up in the last few days, so that will be fixed. Okay. All right, so the natural questions that come out of this now, what are these cell types? What are the gene expression differences between the cell types? And so the first thing you might want to do is find marker genes. So the next step right here, this is a very simple interface. So just simply type in the number of genes that you want to identify per cluster that are most expressed in that cluster versus everything else. Okay. And so this goes through and does a very simple one cluster versus all the other cells. gene expression. And we see for our 6 clusters that I've identified, we have the top marker genes for each one. You can download this list if you like, and that just downloads as an Excel file. We can also, at this point, if we look at the marker genes and we know from literature what each of those marker genes corresponds to in terms of cell type, we can actually go back to the previous window and rename those clusters. So say I did my literature searches, I double checked the marker genes, and I found out that say cluster 0 is excitatory cluster 1. Say this guy, let's see here. Say I found out that these were my astrocytes. Say for kicks that this is my second excitatory cluster. Say I found out that these are my inhibitory neurons. Say this is excitatory 3. And so again, for each of these clusters, using these top G markers, I can make a best guess of what that cell type is. Obviously, there could be further analysis. But let's say I found that out. I can go back to this panel right here, type in what cell type I think each one of these things is. And then if I rerun it, then it'll use that labeling for subsequent steps. Okay. All right, so that's just my guesses right now. And again, this is based on what I've also worked with this data set. And so let's just say, let's take this for gospel for right now. You can use that then if you wanted to visualize each individual gene as well. Say there's a couple of genes on here that you found particularly exciting from your previous research. You can also visualize independently. This will produce a dot plot. And so already you've probably seen these kind of plots in either maybe the supplement of many papers where top marker genes per cell type may be displayed. So that kind of gives you an idea. The dot plots are represented by, I think this is a % of the genes or % of the cells that express that gene. And then the color value is based on the expression of that gene. And then a similar idea with the violin plots. What is that distribution of cells expressing that gene? Okay. And the last step, if people are still hopefully hanging on and doing well, we compare genes and clusters. So this is where I was mentioning before, if you have a single cell data set and you wanted to identify gene expression differences between clusters or cell types, this is where you would do that. So for example, if I was really puzzling on what is the difference between excitatory 1 and excitatory 2, I don't know what these things are exactly, but maybe this will help me get more markers to identify differences. This will identify gene expressions just between those two cell types. And so there you go. So again, we have violin plots comparing them. So we notice that this gene RUNX1T1 really distinguishes these two clusters. I can go into literature. What does that gene do? Does that signify a certain neuronal subtype or whatnot? So that'll help me really nail down what that cell type identification is. You can show the table, you can download the table, so already you can just get that data, just like if you were doing any other analysis, use that table in a publication, and you're off to the races. All right, any questions on the single cell workbench now that we've gone through a nice example? Okay. By the way, if you have a data set that you already have analyzed, so you've uploaded a data set, so you already know what the cell type is, what the t-SNE coordinates are, you can actually start the single cell workbench from this step. So notice that everything we've done up to this point was identifying clusters, identifying cell types. If you already know it at this point, you can actually use, right up here at the top, a stored analysis. And so that would be one of the imported analysis. Again, I don't have one for this one, but it would show up right here. And it would start the single cell workbench right at 5 marker genes. So this is very helpful for a publication. If they already have all that information, you can start the workbench at that point. Okay. All right. For my last portion right here up until break, we're going to just practice building a profile. So you've already seen the major tool of single cell workbench, identifying gene expression differences. I just wanted to walk you a little bit through how do you make plots and how do you produce one of these profiles. So if there's no further questions about the workbench, let's go back to the front page. Okay. And we're going to work with that data set that we just uploaded. We're going to practice a couple of plots here. So underneath your name in the upper right, we're going to click on Dataset Manager. So that's the second item. And so the Dataset Manager allows you to edit and create profiles. And so in the drop-down menu on the left, these are all the different profiles. Again, this is associated with my account, so these you'll probably see fewer profiles on yours. But you can click on an existing profile and edit it, or you can make a new profile, and that's what I want you to do. So right underneath that bar, there's a plus sign. You want to click on that, and you can give your profile name. So I'm just going to say workshop test. I'm going to add that in. So again, if you're building a profile, maybe you're studying data on NeMO Analytics and you wanted to put a couple data sets together, you would do this. If you were designing a profile for your paper, you would go through this as well and kind of tailor that to mirror the analysis that you did in your paper.
Hey, Brian, did we lose you? Brian appears to be frozen on my screen. Oh, yep, we lost Brian. Okay, well, we will assume that he's going to be trying to log back in, so we'll give him a minute or two to do that. We'll just take a little pause. Brian's on his way back. Hey, Brian, welcome back.
Hey guys, sorry about that.
No worries.
And maybe the issues of living out in rural Maryland. So
The workshop was going too smoothly.
yeah, I know.
Something.
Nice job, Brian.
Yeah, very
Thanks.
nice.
Well, it's not the last you'll see of me. All right. Anyway, let's see here. All right. Yeah, let's wrap up the profiles here. So, all right. So, any questions on creating the profile? We're doing well. Okay. All right. So you should, when you search for workshop, you should only see probably one profile. I just have this pre-comp as a backup for the analysis that we just did. But I'm going to look at the Cortex example for workshop, and there's a couple of things that you can do with each profile. The one thing that you initially want to do is simply click on add this to profile. So we're taking that data set, we're linking it to the workshop test profile itself. Okay. And the other options seem pretty straightforward. Remove from profile. We're going to curate in just a moment, so we'll create a plot. While we're here, just go for the other one, change access. So right now, every data set that you upload is private, but you can have the option of making it public right there. And then sharing the data set, I think that gives you a link directly to that data set for if you want to share it with a collaborator. So that's another really nice thing about building a profile. So this also is very helpful for reviewers. If you have your data set up here, they already know that your data is submitted and they can explore the data themselves or sharing with collaborators. Okay, let's click on curate. And stop me if you're, let me get the, okay. All right, so here, very blank page, this is where we're going to add displays. And notice in the upper left-hand side, we have data set type. So primary data is anything that was uploaded with the data. So if we remember back to the metadata files that we added, I had in there cell type information and t-SNE coordinates. That kind of metadata is going to be available through the primary data. So those things that were uploaded with the data set. Stored analysis is going to be anything that was done on NeMO Analytics. So that workbench analysis that we just did would be under stored analysis. So I'm just going to click on that and let's see if we can access those things that we just did. And maybe not. Oh, you know what? Oh, cinnamon sticks. Why not? Oh, okay. I may have forgot to tell you guys to push save. I think that's the issue. So if you got, and I deeply apologize for this, I did forget a step back here with the workbench. Pressing save is the thing that, that really unlocks adding that analysis to the data set. But don't worry, we have some canned analysis that we can work with here. So primary data, if we go back to that real quick, you can choose a display type. And there's a couple of things that we can do. Scatter plots are nice and flexible. As we saw with the Murfish, you can use any and all XY coordinates if you want. This is also helpful if you wanted to plot, say, like gene expression versus a principal component. Anything that's within the metadata and in your data, gene expression itself can be analyzed in the scatter plot. There's two different ways of displaying the single cell data. t-SNE static, what that simply means is you can put those plots together like we saw in the profile. So you have a gene expression plot and a static plot showing just those cell type information. The dynamic is simply a t-SNE or UMAP where that just shows the gene expression data. A bar plot, violin, and line, again, it's if there's anything available to you, I like to split up for the bar plot, gene expression versus cell type, same thing for the violin. So there's a lot of ways that you can dissect that data and display it in new ways. What we're going to take a look at is the t-SNE static. So that's the second option right there. And I know in this data set there's the gene SST. So that little neuropeptide. Again, that's not hugely important, but it does unlock the next steps. And for the display parameters, this is going to give back to me those cell metadata that we used in the upload. So for my XY coordinates, I'm going to use t-SNE1 for the X and t-SNE2 for the Y. So again, this was a pre-computed t-SNE. And already we see that gene expression. So these are always side-by-side plots. We have gene expression versus, and then you can add in a colorized legend. So that little click box right there, and I can colorize this by one of those metadata fields. We have cell type from my previous analysis, and that's going to add that in right there. So that looks a lot like the profiles that I showed you before for the mini atlas. Things like plot by group are good for, I think, the bar plots and violin plots. So you can, each one of these things has its own little help icon here, and you can explore the different plotting types. Let's go through. Okay. And scrolling down to the bottom, you can save the display. It gets us back to this point. You can make as many plots as you want. It's just that whichever one you want to have displayed in your profile, you're going to say make default, and it is there for you to use. Okay. Any questions on plotting? Okay. Yeah, with the gene names, it's pretty flexible. It'll always try and match that back to the ensemble ID in the background and display the correct names for you. Okay. I think the next step, let's go to the back to the dataset manager. And I'm going to further refine that profile. So selecting again workshop test, I now have my data set within it. I've curated it. I've made a plot for it. You can also adjust the arrangement. So if we have multiple data sets in here, let's just, if you want to find any and all other public data set, let's just say if we type in motor cortex, we can try and find another data set that may be already curated. And this is where you can really explore NeMO analytics, look at the data sets that are there, find ones that make sense for comparing. So I'm just going to grab this one since we're running out of time here. But that's now just added to my profile. And if you want to change the display of how the data sets are arranged, you go up to the top here, click on arrangement, and you can actually take these windows and expand them. So these things, it's a 3 by however long profile. So I've just simply expanded that plot, how much space it's allowed to have. I'm going to do the same thing for this guy right here. And I'm going to save that layout. So that step is really where you can kind of reconfigure the layout of the different data sets. Okay. And then I think for our last great step here, let's go back to the front page. And I'm going to take a look at that profile. So workshop test, I've clicked that into my profile. I'm going to look at, again, that gene SST. It could be anything, but that one data set only has a few genes in it. So and there you go. And so what I've done here, I've taken 2 very disparate data sets, one that perhaps I've just generated, and I'm comparing it now with any and all data sets that are within our ecosystem. And so I pulled up this actually it's kind of a nice comparison right here. This well-curated data set from the Kriegstein lab looking at early human development. And you can make that direct comparison like, okay, I know that this gene is a strong marker of this grouping right here. It probably corresponds to this grouping that this other publication has really looked at, really thought about a great deal. And I can make these nice comparisons across data sets. So hopefully I've explained to you the flexibility of the profiles, a few of the tools that are on NeMO Analytics. Again, I can't emphasize enough, this is a very good platform if you're publishing a paper, you want to put your data on here as well. And you can put those links in your paper and then people can really explore. They can see the figure in your paper and then jump right into the data itself. So there we go. So thank you very much for your attention. And again, we're also very open to brutally honest feedback on the site. We're going for a little bit of a reconfigure about the navigation of the whole thing. So this will be a good time if you have, if you're excited about a tool, but you're just wondering about how best to navigate in the site or improvements to the site, please feel free to email us, and we'll definitely take it to heart as we're improving the site over time. So thank you.
Michelle or Anoop, are you there?
Yeah, so just giving a couple of more minutes. Otherwise, I believe there's a scheduled break between now and 2 o'clock. Yeah, Michelle says she just lost her connection. So, yep. So we have a break between now and 2 o'clock. If people don't have any questions, let's reconvene around 2 and we can pick it up from there.
All right, folks.